The year was 1909 when a Danish chemist needed a way to express how acid solutions influence proteins. So he developed the pH scale. This chemist used logarithms as a simple way to quantify the number of hydrogen ions in a solution. Søren Sørensen was the name of this chemist, and he used well-established properties of elements to calibrate his new tool, such that 1 gram of hydrogen as an ion, also known as hydronium, is equivalent to 1 mole of hydrogen, which equals the famous Avogadro's number of 6.022 times 10 to the 23rd atoms. Sørensen calibrated his scale at 0, which tells us a 1 molar solution of an acid that dissociates into hydrogen ions would have exactly this huge number of ions per gram of the element. With numbers this large, adopting a logarithmic scale makes sense. Using a linear scale would simply be unmanageable. Most scientific calculators have a dedicated button for calculating logarithms and inverse logarithms. Both functions are needed to take a value from Sørensen's pH scale and convert it to molarity, a measure of how many ions are in a liter of solution. Sørensen used the negative log of the hydronium concentration as his definition. So for a 1 molar solution of hydrochloric acid, the negative log of 1 equals 0. Sørensen's pH scale ranges from 0 to 14, but it is possible to have an even more concentrated solution of hydrogen ions, such as a 2 molar solution, in which case pH would be calculated as a negative number. The negative log of 2 equals negative 0.3 in this case. At the high end of the pH scale, the hydronium concentration is just a tiny fraction because the solution is overwhelmed by OH ions. Even without adding other molecules, the water molecules will auto-ionize just due to the properties of H2O, where the molecule can either donate or accept a proton, which is equivalent to hydrogen, an element that consists of 1 proton in its ionic form. Thus, at any given moment, some of the ions in the solution will be hydronium, while others will ionize to OH. When the hydronium concentration equals 1 times 10 to the minus 14, this comes to 14 on the pH scale, and we know we are working with a strong base. At 1 times 10 to the minus 7th, we say that the pH is neutral at a pH of 7. When we measure the pH of a solution with a modern sensor, like this model, the device detects tiny voltage differences through the glass tip of the electrode, and the millivolts, a unit 1,000 times smaller than 1 volt, relate directly to the concentration of hydronium ions. When you think of examples of acids and bases in your everyday life, what comes to mind? Do you think of lemons for acids, or maybe vinegar? And for bases, maybe bars of soap or window cleaner? We can verify where these or any items fall on our pH scale with a quick measurement. Soil solution from my garden typically test at a pH of 6.5, a value that strongly influences which micronutrients plant roots can take up from the soil. Plants become deficient quickly when soil pH is too low or too high. Our bodies produce and process all sorts of fluids and molecules that can be classified as either acids or bases. And the chemistry of our body tissues is such that it must be kept within a very narrow range. This is known as acid-base homeostasis. How narrow? Well, nerves cannot fire properly if they deviate from a pH of 7.4. Decreases in brain pH are a symptom of neurodegenerative diseases such as Alzheimer's and Parkinson's. Blood pH must stay between 7.35 and 7.45. Below 7.35 in a person is an acidosis, and this may cause a racing heartbeat, dizziness, confusion, and labored breathing. Acidosis is common in people with uncontrolled diabetes or those experiencing kidney failure. Above 7.45, a person is an alkalosis, and this affects heart rhythms and brain function. Muscles may cramp or spasm. Alkalosis may be caused by dehydration or severe electrolyte imbalances. If you hyperventilate long enough, perhaps from a panic attack or an anxiety-related disorder, you can induce alkalosis by expelling too much carbon dioxide too quickly from your body. You will become dizzy and lightheaded and may even pass out. Brain seizures are also possible. Obviously, we need to respect these narrow pH limits, and it's worth considering how our many habits surrounding food, medicine, and exercise complement this evolved physiology. Our kidneys provide long-term pH control by filtering the blood and selectively reabsorbing important electrolytes such as sodium, potassium, magnesium, and calcium. We get shorter-term pH control by breathing. As mentioned with the example of respiratory alkalosis, ventilating our lungs faster raises blood pH. When we are stuck in long, boring meetings at work or in poorly ventilated rooms, our breathing naturally slows, causing blood pH to decline as carbon dioxide builds up. Neuroreceptors in our brainstem, as well as in arteries like the aorta and the carotid arteries, detect pH changes in the blood and signal to our breathing muscles to inhale deeper and faster. These are all unconscious physiological responses, and it's a good thing too. A variety of molecules contribute to the pH buffering in our blood. A buffered solution is one that resists rapid changes in pH. So with the narrow homeostatic range for acids and bases, buffering is crucial to keeping us alive. Bicarbonate ions are the primary buffers in our body, and these ions react with hydrogen to form carbonic acid. And the reaction is reversible to release hydrogen. These free-floating hydrogen ions are what we can measure as pH. And of course, they are what also changes the structure and function of cells throughout the organism. As our kidneys produce urine, bicarbonate ions are dynamically regulated, such that urine is usually acidic. Plasma protein like albumin can also buffer our blood by binding and releasing hydrogen ions, as does hemoglobin inside our red blood cells. An enzyme within the red blood cell, known as carbonic anhydrase, reacts quickly with carbon dioxide to form carbonic acid. The hydrogen ions released during this reaction are scavenged by hemoglobin, which release their oxygen molecules. Oxygen unloads to working tissues while carbon dioxide gets hauled away. Our red blood cells are also making bicarbonate ions from the carbonic acid. So, in a sense, the carbon dioxide produced by our cells as a byproduct of harvesting energy from food is not entirely waste. Some of it gets converted into bicarbonate, and that bicarbonate is used to help stabilize blood pH. What does it mean mathematically for blood pH to span the acid-base homeostatic range? If blood pH drops from 7.45 to 7.35, it means the hydronium concentration has increased, but by how much? We can summarize it with this equation. pH is equal to 7.45, which is equal to the negative log of the hydronium concentration. Multiply both sides by -1 so our log is positive. We can then take the inverse log of each side of this equation and solve for the hydronium concentration. And that gives us 3.55 times 10 to the minus 8 moles of hydronium per liter of blood. If we do the same calculation for the other end of our range, pH is equal to 7.35, that gives us a hydronium concentration of 4.46 times 10 to the minus 8 moles. The hydronium concentration has increased nearly 26% with this seemingly small shift in pH. Any solution that changed one whole unit of pH would mean a 10-fold increase or decrease in the hydronium concentration. And in most biological systems, a change this large would be profoundly disruptive. Hopefully now the utility of Sorensen's pH scale is clear, as it gives us a great tool for comparing physical quantities of hydrogen in a solution. Sorensen's tool can be applied to any kind of ion, such as OH or nitrate. pH is an essential parameter for understanding human physiology and basic chemistry, but also things like industrial manufacturing, as well as the health and functioning of soils and aquatic ecosystems.